Make sure you are dressed to harvest treated fruit by wearing a shirt with long sleeves, a coverall, socks, shoes, and chemical-resistant gloves. Do not use the same gloves you wore while spraying the test substances. Use a different set of gloves for the harvest. It is recommended that you write on the package of one set of gloves that it is meant only for the harvest. Residue samples will be collected at 3 time intervals: 1 hour after the application, 24 hours after the application, and 72 hours after the application. Make sure that you have already harvested the tomato fruit from the untreated control plot before you spray the treated plot. Wearing gloves, take fruit from the top and bottom of many different plants. Collect at least 12 mature fruit, weighing at least 1.5 kilograms in each of the 4 sample bags. While red fruit is preferred, all of the fruit does not need to be red. The fruit may be mature and still appear green. You should do this by walking up and down through the plot 4 times. Collect fruit as you walk through the plot. Complete this procedure 4 times. Do not collect fruit from the ends of the plot or the 2 outside rows. In other words, collect fruit only from the 2 middle rows while avoiding the ends of the plot. Put the fruit in the ir4 plastic-lined sample bags you received with the sprayer. Make sure each sample bag is labeled properly with the correct date and sample ID number. You should collect a total of 4 samples from the treated plot at each time interval. Collect 4 samples 1 hour after the application. Collect 4 samples 24 hours after the application. And collect 4 samples 72 hours after the application. Notice that the sample bag is covered with a plastic bag so that the outside of the sample bag doesn't get contaminated by touching the treated plants. Place the 4 sample bags in the treated sample ice chest until you can put them into a freezer. Monitor and document freezer storage temperatures every day until the samples are shipped. Use continuous temperature monitor devices if available. Try to put the samples in a treated sample freezer as soon as possible. The field site should be near enough to your freezers so that it takes no more than 4 hours from collection until the samples are placed in the freezer. You should place treated samples into a different freezer than the untreated freezer. If separate freezers are not available, contact Michael Braverman. Five days before shipping the samples, contact Michael Braverman by phone or email for final directions. Only ship samples on a Monday or Tuesday. You will be provided with sample boxes for shipping the samples. Tape the bottom of the boxes securely. You can also place labels on the boxes. Make sure each box has a dry ice label. Follow all shipping regulations required by your country. It is very important that you keep the treated and untreated samples away from each other. Write on the box, "Frozen research samples, for research purposes only, not for human consumption." Use the residue shipping form and chain of custody form. Make a copy of each form for your records. Then place the forms into a plastic bag. Tape the plastic bag to the inside lid of the box. Use separate boxes for treated and untreated samples. Use express shipment. Put the samples in the sample boxes with a ratio of at least 4 kilograms of dry ice for each kilogram of sample. Blocks of dry ice are better than small cubes. Do not use regular ice. Seal each box shut. After packing, place the boxes back into the freezer if possible until they are ready to be shipped that day. Do not keep the samples out of the freezer any longer than necessary. If you are transporting the boxes in a truck or car, turn on the air conditioning and cool down the vehicle before placing the samples into the vehicle. 2 samples, A and B, from each treatment and sample date should be shipped to the location specified in the protocol. Keep the other 2 samples, C and D, as a backup in case the primary samples are lost or destroyed in shipment. Don't forget to document everything you do in your raw data book. This will help to maintain the integrity of the study. When all samples have been shipped, make a photocopy of the raw data book and send the original to Michael Braverman. Keep the copy for your records and security. In this film, we have described the main parts of the study, but we did not cover all the details. Please be sure to thoroughly read the protocol and field data notebook so that you become familiar with the types of information you will need to collect and record. And remember, if you have questions, don't hesitate. Contact Michael Braverman. He will be happy to answer all of your questions. Thank you for participating in this global residue trial that will benefit growers throughout the world. Let's review some common errors and how to avoid them. One of the most important things to avoid is contamination, such as handling fruit or walking through the treated plot and then walking through the untreated control plot. Do not contaminate the sample bag by allowing it to come in contact with treated plants or soil. Cover the sample bags with an outer plastic bag so that the inner sample bag remains clean. Avoid errors in measuring or mixing the pesticide and water. Avoid errors in application or walking speed. Do not make an application during high winds or just before rain, because the test substances will be washed away by rain soon after application. Avoid putting treated and untreated control samples together during collection, transport, freezing, or shipping. Avoid delays in getting the samples into the freezer. These are research plots, not tomato gardens. Do not allow others in the research plots, and don't eat or give away samples. This is only a partial list of common errors. If you are in doubt, please contact Michael Braverman. He is the director for this study. If you have questions or need directions about anything that you plan to do or have already done, please contact Michael Braverman at braverman@esop.rutgers.edu.